Hello students, so today we're going to start with a new chapter, Heredity and Evolution. Heredity. It is also called as inheritance or biological inheritance. It is the passing on trait from parent to the offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parent. So the study of heredity involved or the study of heredity in biology is called as genetics. So we're going to discuss brief about heredity and variations. Heredity, the transfer of character or trait from the parent to the offspring is called heredity. So the transfer of character from parent from one generation to another generation is called heredity. Variation. The difference between the characters or trait among the individual of the same species are called as variation. Next we are going to discuss accumulation of variation during reproduction. When organisms reproduce, the offspring show minor variation due to inaccuracies in DNA copying. So these variations are less in asexual reproduction and more in sexual reproduction. So in asexual reproduction, what you can see, there will be the actual parent copy. Okay. So in sexual reproduction, you cannot see the actual parent character, but even eventually you can see the more variations occurs in sexual reproductions. Some variation are useful variations and they help the organism to adjust to the changes in the environment. So some variation do not help the organism to adjust to the changes in the environment and they may die and become extinct. Accumulation of variation during reproduction. Variation in organism mainly occur during sexual reproduction because of mutation caused by error in DNA replication. Thus no two offspring are identical to each other. Other cause of such variation or crossing over during meiosis, that is cell reduction, separation of chromosomes, during gametogenesis from both parents at the time of fertilization. Hence we see that each generation provides the next generation, but some variation through the body design remains the same. So some new variation are also produced in the new generation too. The variation keep on accumulating and passing to more individual in each generation. Thus, as we proceed to later generation, the frequency of variation goes on increasing. Now, we next move on to the rules. The rules for inheritance of character. Okay, so here the character which is refers to trait. Characters are transferred through gene present in the DNA molecule and the chromosomes present in the nucleus of the cell. So where the DNA molecule present inside the chromosomes, where the chromosomes present inside the nucleus, and where nucleus is located in the center of the cell. The inheritance of character is due to the fact that both the father and mother contribute equal amount of genetic material of the child. So here the genetic material which is referred as DNA. So for each trait there are two factors, one from the father and one from the mother. So there will be like 50-50% to make 100% of the young ones. So the haploid gamete, that is N plus N, it produces a diploid 2N. So Gregor Johann Mendel conducted an experiment with garden pea plant and determined the rules for the inheritance of trait. So first we see about the DNA. So where the DNA present. So here you can see the organisms which have cells. Inside the cell there is a center located with the nucleus. So inside the nucleus there will be the chromatic network called the chromosomes. So inside the chromosomes it is binded with the protein histones. So inside that we can see the DNA. Okay, so DNA. So inside the DNA there will be the gene which codes for the protein. Okay, so this is another snap of where you can correlate the human cell to the chromosomes to the DNA. Structure of DNA. Actually, what is this DNA? Deoxyribonucleic acid. It is an expanded form. It was first isolated by the scientist Frederick Miescher from the nucleus of the pus cell in 1869. He named it as a nuclein or nucleic acid because of its acidic nature. So DNA is the carrier of genetic information from generation to generation. It transmits the hereditary character in a coded language from parent to the offspring. DNA is a macromolecule or a polymer. It is made up of a very large number of nucleotide units and hence is termed as a polynucleotide. Each nucleotide unit in a DNA molecule is made up of three components: deoxyribosugar, nitrogenous base and phosphate group. So first we discuss about deoxy sugar. It is a pentose sugar represented as S. Nitrogenous base. Each nucleotide unit has a nitrogen containing bases. They are four type. Okay, they are the four type. They are adenosine, thymine, guanine and cytosine. So according to the nitrogenous bases are arranged in a DNA molecule or based on two types. They are purine and pyrimidine. So the purine in the DNA molecule are adenosine and guanine. Pyrimidine in the DNA molecule are cytosine and thymine. Next we move on to phosphate group. The phosphate group contains one phosphorus atom and four specifically linked oxygen atoms. Thus, there are four types of nucleotides in a DNA molecule depending upon the kind of nitrogenous base present in each nucleotide. I repeat, the nucleotides are adenosine, guanine, thymine and cytosine. So, there will be the difference between DNA and RNA. So, the major difference will be DNA is a double-stranded and single-stranded will be a RNA. And another one major difference is, so here the adenosine is bonded with thymine, cytosine is bonded with guanine. In RNA, so in place of thymine, RNA is replaced with uracil. Okay, so in place of T, the RNA which have a single strand is replaced with U, that is uracil. Thank you.